really very happy that Kira is here, and telling about your obstacles to come here fills a complete evening of interesting discussion and conversation. So it's really great, Kira, that you're ready and that you're here today. So I think everyone knows her. Kira did her bachelor in Stanford, and then she moved to the University of Cambridge and did her PhD with Peter Lally. And I don't have the exact years in mind, but something
long time ago.
Long time ago. And then she also moved to Saarbrücken. This was, I think, 2007 around. And this is for sure, and also from 2010, as she has a professorship at the University of Lorraine University in France. Probably, I mean, we have all read her papers, and very likely we have all cited, for example, her Millennium Review, which is cited 1,700 times, which is scholar, which is, I think, the most highly cited review in the field. Kira did a lot on domain-domain interaction, docking domains, protein-protein interaction, structural studies, so always a lot of structure involved, NMR, X-ray, SAX, all what you wish to have. So thanks for coming again, and then the stage is yours. It's really a stage here.
Literally.
Yeah. So, yeah, please.
Thank you very much for that super introduction. And of course, for the invitation to present some of our works today at this wonderful conference. It's so nice to actually have a conference for the first time in a long time, for me at least. Despite the best efforts of both the SNCF and Deutsche Bahn, I made it yesterday and even set fire to the tracks, literally, to keep me from coming. So, and thank you, Nadine, for taking my place yesterday. Okay, so today I'm going to tell you two related stories, which I think illustrate how we can apply structural function relationships to genetic engineering PKSs, but are also cautionary tales in the sense that they illustrate some of our major limitations to understanding these systems, which continue to negatively impact the efficacy of synthetic biology. So I'm going to start, because this is rather an expert audience, by a thought exercise. And we're going to try to do some docking domain-based engineering. So of course, docking domains, as you all know, are the short sequences at the extreme termini of the PKS subunits that mediate contacts and do this specifically. And so what So the idea here is to take a model system, which consists of 3 subunits, 5 modules, and to carry out a module swap here. Okay, because modules are emerging as quite useful biobricks for this type of experiment. And at the same time, we're going to split the subunit 2 into 2 subunits, because this can dramatically increase engineering efficiency. This has been demonstrated as an approach in both PKS systems and in RPS systems, or recently due to work from Helga. But in order to do this type of experiment,
you maybe have to go a bit closer when you
okay.
We have so many signals here, this is the problem.
Okay, I'll stand. What it would be ideal to be able to do is to analyze bioinformatically all of the subunits in their termini in order to identify the docking domains that are present. And so we need to know, in fact, what their extremities are and to which structural types they belong. And this is in order to both maintain the native docking interactions between subunits, the new subunit 3 and subunit 4, and to introduce a novel interaction, which is intrinsically orthogonal to the others. Okay, so this won't necessarily overcome all potential blocks in terms of protein-protein interactions and substrate specificity, but it will, at least in theory, ensure a specific ordering of the subunits in the engineered system. So I'd like now just to remind you about the docking domain classes that have been described to date. So there are 3 from the cis-AT PKSs, as shown here, And we're going to see 1, 2, and 3 again in this talk. And there are 2, which are known from transit diffuses to date. Okay. So I'm not going to go into the details about these structures, but just point out one important feature, which is that they are intrinsically orthogonal to each other. That means that they don't interact. So as a test of our ability to carry out this type of experiment, we chose the N-siloxane mixed PKS NRP system. Why? Well, it has 7 polyketide synthase subunits, which is a relatively large number. And so there are 6 distinct PKS-PKS interfaces. And it is also only one of 3, to my knowledge, systems in which cis-AT and trans-AT PKS subunits are mixed together. And so we were interested to understand how docking occurs at this hybrid interface, because the ability to create such novel junctions would obviously be useful in the future. During the course of this work, the Chalice group published the characterization of this interesting NRPS interface here, which is involved in mediating the unusual offloading mechanism of N-siloxane. And here they identified, it's called the type 3 docking. Now, to start out this analysis, there's a compatibility problem here. Too bad. To start out this analysis, we carried out what I would call non-specialist sequence analysis, a very standard application of clustal, for example, of our docking domains. So we identified the extreme functional domains in each of the subunits. So this was typically ACP and KS. And then, of course, the flanking regions could potentially contain docking domains. And then we just carried out simple sequence alignment of those regions against the known classes of docking domains. So bona fide and putative members of each of the other classes. And so what was supposed to be clear, but isn't from this slide, is that the type 1A docking domains, shown here, form a quite unified class, and this is backed up by Marnix's research results. Whereas our N-siloxane docking domains, so these are supposed to be highlighted in red, they're all here, they show some sequence similarity to the type 1A domains, but they're mostly similar to each other. And so whether they belong to this group or not was inconclusive. And we had the same result when we compared these docking domains to the other sequence classes. And so we couldn't, on the basis of sequence analysis alone, even make a confident prediction of what type of docking domain we had. And so we decided to carry out characterization directly of all of the interfaces in the system. Now, because we couldn't identify the classes, we didn't know the extremities of the docking domains, and so in fact we had to express and characterize multiple versions of essentially all of the docking domains in the system. We then screened for the physiologically relevant versions of the domains by comparative ITC analysis, using as a major screening criterion those interactions that gave the highest affinity. And in this way we could show that we had an interaction at 5 out of the 6 interfaces in the system. And the affinities were within the range of those previously described for docking domains, so that's all very reassuring. And the only exception was for this unusual split modular interface between these 2 subunits, in which there are flanking KS and parent domains. Now, the docking domains that we identified passed another critical test, which is that the interactions were specific. So we could show by a series of cross-dock experiments that there were essentially no interaction between mismatched pairs. So this was all very reassuring. Next, we carried out a battery of biophysical tests available in the laboratory to characterize both the individual docking domains and the docking domain pairs. So for example, by CD analysis, we looked at the secondary structure of the docking domains, and we could show that while the N-terminal docking domains were at least partially structured into alpha helix, so we have these very characteristic spectra, the CDDs, in contrast, look like random coils. And so this led us to wonder whether in fact the CDDs are what are referred to as intrinsically disordered regions. So these are protein sequences which only adopt stable folds in the presence of their partners. And so to investigate this question, we carried out what we call the double cuvette experiment, where in essence we could compare the combined spectrum of the individual docking domains, so they're not together in the experiment, with the spectrum of the DDs, which are mixed. And because we can convolute the spectra to determine, for example, the percentage alpha helix which is present, we can show that, in fact, structuration increases when we combine the two together. And so this is evidence, albeit indirect, that the C2 docking domains are becoming folded when they interact with their partner. And so indeed, we could show that for the 5 interfaces where we saw an interaction, that we saw an increase in structuration consistent with the IDRs becoming folded. Okay, so the one exception, again, the split interface, where there's no interaction between the docking domains themselves. Ooh. Wow. I must have done something.
We just scratch it for you.
This is the kind of feedback I'm having. Yeah, the one with the red block there. Yeah. Okay, so just to put this together. Okay, so summarize the data on the docking domain. So it's amazing that you can put the data from essentially a whole PhD on one slide. So what we showed overall is that all of the CDDs were uniformly monomeric and showed convincing characteristics of being IDRs. And this is the first time, in fact, IDRs have been described as operating in cis PKS systems, whereas the NDDs were, with one exception, homodimeric and well folded into alpha helix. So we put together these biophysical data along with supervised realignment of our sequences, because now we knew the physiologically relevant extremities, in order to assign putative, and I insist on that, putative docking types to each of the interfaces. And so what this revealed was that we had 2, what I refer to as type 2 related, and I'll tell you why they're related in a moment. 2 type 2 related interfaces, here and here, and 4 putatively type 1 related interfaces. And so combined with Greg's work, what we showed here is that in fact there are 3 structurally distinct types of docking domains which are employed in this one system in order to achieve specificity. So there are multiple interfaces which are potentially mediated by the same structural class of docking domain, but in fact none of these interfaces is identical. So we have, for example, potentially differential electrostatic drivers of association of the docking domains as expressed by these divergent delta PIs, and differential contributions of the flanking domains to these interactions. And so we think that this is another layer of specificity in the system. Now, to shore up this result, we decided to use NMR to solve the structures of 2 complexes of docking domains, and one of each type. So the first one here, at this junction where we actually had an interaction, and the docking domains operating at this interesting cis-AT trans-AT PKS interface. So I've shown here, in fact, the NMR structure of just the N-terminal docking domain. So it's shown in blue and silver. And it's superimposed with the founding member of the type 2 docking domains, which is pure H and D. And so what you can see is that we have in both cases a 4 alpha helix bundle, but that the docking domain in the N-siloxane system has a different topology. And so this alters, in fact, the surface properties of this docking domain, which lead to the coupled folding and binding of the C-terminal docking domain partner. And this leads ultimately, although we obtain an 8 alpha helical bundle, which is characteristic of the type 2 docking domains, the overall shape is different. Okay, so we have something that's similar but not the same, And so we refer to this as type 2 related. So in the case of the interface here between the cis and the AT, cis-AT and trans-AT PKS subunits, we have a structure which rather closely resembles a type 1A document domain, with the notable exception that this homodimerization region is completely missing. So in fact, this sort of minimal document motif is capable of mediating communication between the two large classes of modular PKS. But if we look a bit closer at the interfaces, what we see is that the charge-charge interactions, which potentially mediate specificity, and other interactions which are involved in stabilizing the document domain complex, and this is just a subset of the interactions that you see, look to be hybrids between the two major classes of type 1 document domain, 1A and 1B. And so again, what we have is something that's overall similar, but different. And so we call this type 1 related. So what I think our work does, in fact, is to raise important questions about document types and subtypes. And this situation arises because, in fact, these classifications are based on the structure elucidation of one or at most two document domain structures. And so we really have no idea what sequence diversity is allowed within a conserved document domain architecture. And what we've shown, in fact, is that sequence divergence can manifest itself in several different ways. So, for example, in subtle modulation of the electrostatic drivers of association, or residues contributing to stability, or in more dramatic structural alteration of the document domain complexes. And so I think as a community, we have quite a lot of work to do in order to better define what document domain types exist. And this will ultimately allow us to establish the structure sequence relationships that are necessary to allow for confident prediction, just based on sequence analysis alone, of document domain types, and maybe even of their substrate specificities, or their document specificities, sorry. In the meantime, we have a useful and ever-growing toolbox of document domains, so recently boosted by the lovely work from Martin and Helga's lab on syn-zip domains, which will hopefully allow us to explore the full scope of module and subunit-based engineering. Now, another way to carry out engineering based on modules is to actually covalently fuse modules together. And so we've been exploring this strategy to try to rationally alter stereochemistry in a monopoly potential-based system. Now, this work is again based on structure-function relationships, notably from Adrian Keating-Clay's lab, and in particular on the ketoreductase domains. So these domains catalyze ketoreduction, the beta ketone group. And there are several types of ketoreductase domains. So the A1 and E1 catalyze simple reduction to give the two alternative stereochemistries that they position, whereas the A2 and B2 domains catalyze in addition and polymerization at the adjacent methyl center. And so in principle, by manipulating a KR domain, we could access two stereocenters at the same time. So initial work in this direction was carried out in Peter's lab quite some time ago in the Depth-1TE model system, which I think everybody knows. And so what was done was to replace initially an A1 KR domain by a polymaker region. And then to insert in place of that KR domain a panel of A1, A2, E1, and B2 KRs. And what this experiment showed was that you could replace an A1 with other A1s, and this worked quite effectively, and also with B1 KRs. Okay, and so here you get a change in the hydroxyl stereochemistry, sorry. But all attempts to introduce A2 and B2 stereochemistry only yield traces of the expected products at best. And so there was something about polymerizing that methyl center which seemed difficult. So in more recent work from my lab, we tested, in fact, a much more extensive range of A2 and B2 KR domains. And so we found a few, not a lot, but a few, and notably from polyene PKSs, which gave the expected goodness gracious, which gave the expected product in respectable yield, I would say, okay? But the stereo control wasn't 100%, and in some cases, the ketoreductase didn't act at all. In the case of the B2 KRs, the result was even more discouraging. So again, only traces of the expected product in which you have a change in methyl stereochemistry and hydroxyl stereochemistry was obtained. And so what this led us to consider was perhaps there was something about an intrinsically A1 modular environment that was prohibiting these KRs from acting correctly. And so to address this, we decided to carry out a whole modular exchange. So exchanging a KR1 type module for A2 and B2 equivalents. But of course, in order to do a module exchange, you have to grapple with the basic definition of the module. So classically, modules are defined as running from KS through any processing domains that are present to an ACP. And in fact, we find many modular subunits which have precisely this domain composition. However, there is an alternative definition of modules, and this is suggested by the coevolution, of KS domains with their upstream processing domains, at least in certain modular PKSs. And so this suggests, in fact, that modules should start with an AT, go through the processing domains to the ACP, and then terminate in the KS. So in fact, module exchanges have been carried out with both of these modular definitions and have worked to some extent. And so it wasn't clear to us which one of these options we should use, and so we tried both. So in terms of the whole module swap, we used a site at the conserved N-terminal end of the KS domain. And for the tri-domain swap, we use one at the conserved C-terminal end of the KS domain. And so the difference between these two is essentially a KS swap. In terms of the site at the end, it was the same in both cases, and right at the end of the conserved ACP domain, thereby maintaining, in fact, the ACP-TP linker intact. So here I've shown the results, so this is about half of a PhD. It's a huge amount of work for a few data points. And I'm just going to summarize the major results for you. So what we showed is that for the one case in where we could compare, in fact, directly the module swap and the KR swap, that the module swap gave really dramatically improved overall yields. And although there was a minor loss in terms of stereo control. Now, in terms of the interesting question about which modular definition worked better, it turns out that the classical definition in each of the cases that gave the target product was better than the alternative module, both in terms of overall and relative yield. Now, another notable observation from our results is that we in fact tried quite a lot of modules, and some of them failed to be. And the notable difference between the modules which worked at all and those which didn't, was that the modules that didn't work came from the N-termini of their respective subunits, whereas those that did work were sourced from an internal position. So in terms of the B2 type modules, again, we could show that the full module swap relative to the equivalent KR exchange worked better. But this time, it was the tri-domain. So the alternative module definition that gave the better result. But again, we saw that N-terminally sourced modules essentially didn't work, and an internal module did. So just to conclude on this part of the talk, so unfortunately, we don't have the answer. So what is the domain composition that constitutes a functional module? Okay, it appears to be mixed. And fundamentally, when we carry out module exchanges, we always introduce at least one non-native interaction. So for a whole module swap, we maintain the native KS-ACP chain condensation interface, but we introduce a non-native chain transfer interaction, and it's not necessarily the case that KS substrate specificity will be adapted to the substrate that it sees from the donor module. In the case of the alternative boundaries, the chain transfer interface is native, and the substrate specificity of the KS is intrinsically adapted to the incoming substrate, at least at the alpha positions. But what we introduce is a non-native KS ACP interface. And so overall, what our data show is that how detrimental these non-native interactions are to the functioning of the hybrid PKS can vary depending on the source module. And so I think from a pragmatic point of view, at least for the time being, we should try multiple boundaries when we're carrying out this type of experiment. But our data also show that perhaps we're going to be more lucky in terms of results if we use modules that are sourced from internal positions relative to those which are located at the end termini of their source units. Thus concludes my talk. I'd like to thank the wonderful students who I've shown here who carried out this work, Andy Risser, Jean-Malo Nassauer, and Thibaut Benivon, my excellent colleagues, these are my permanent colleagues who make up my team, members of the IBS Lore platform who contributed to the biophysical analysis, and these various funding bodies. And thank you very much for your kind attention.
Hi, Kira. Thanks for a really wonderful talk. Just on the final points you were making there about how modules are defined, I think there's a third way that we should think about, and that's how nature defines modules. Because I'm pretty sure that in these systems, in the fungal systems that we work with, that recombination is the way in which new synthases are formed. And recombination is going to happen at the sites of highest homology. Nature doesn't care about the domain boundaries that we see. I think probably swapping at the positions of the highest homology will introduce the fewest breaks, damages to the system, and are more likely to work.
So the question is, why not follow nature's guide as to where natural recombination sites occur? And I think that's a very good point, and that goes along with the isomerase work where they induced recombination spontaneously, and they found, in fact, that recombination sites, at least in the rapamycin system where there's a very high overall mutual degree of similarity, that those largely occurred within the most conserved regions of the KS and AT domains, notably. So in fact, we've been inspired by that result, and we've tested a couple of sites which they identified. And we saw an improvement in our hybrid system, but it didn't solve all the problems. So I think that there remain other substrate specificity issues which also contribute to the low yields. So it's maybe not a panacea.
Thank you for the nice talk. Can you guess what kind of role the PKSs plays a role in the specificity of this unusual substrate?
I'm not sure to understand the question.
What kind of role the PKS plays in the unusual substrate and the yield?
If it biases the yield, you mean?
Yes.
I don't think it's a major contributor. Based on my own work, actually, as a PhD student, I tested the effects of all 4 diester isomers of the alpha and beta positions on thioesterase activity. And there was a minor difference between them, but nothing that would really, I think, contribute to the differences in yield that we see.
In your one split module, module 3, so are you sure that you have a docking domain there? So would it be possible that you do not need a docking domain just because you have a big enough interface?
Absolutely. So we have sequences that show, again, and it's not definitive because we don't have the structure, similarity to, in this case, type 2 docking domains. But what remains possible for me is that there's some other type of docking that's involved. So, for example, a docking domain interaction, as they showed in transient PKSs, or that the modules, the domains themselves, the flanking domains interact with each other. There's also, in that case, downstream of the KS, some, I think it's 200 amino acids, if recollection serves, which don't show a high homology to anything. So there's something else in there which potentially is contributing to the interaction of that system at that point. So it's not clear whether the docking domains are involved at all. Any questions? Okay, since I'm the chair, I want to ask myself a question. Oh, you have a question?
I have a question soon. I'm switching on the microphone. So,
No. I have to read it. Yeah. It's from Adrian. Adrian says nice work. Have you tried swapping the central module of depth P? Have you tried swapping the central module of depth 1P? The central module, probably that's module 1, as far as I understand. So you have this, the depth 1 is the LB and 1 and 2P.
No, we haven't tried.
That's, I think, the question.
Yeah, no, because we wanted to compare directly with all the previous literature on the KR swaps. All of those were carried out in module 2. And also in that way, you only have a TE downstream, and so you have a less complex system to analyze. So if there's a subsequent chain extension module, then you bring in all of its substrate specificity and etc. So to simplify the system, we focus on module 2.
Okay, yeah, that's this.